the first paper we published on that subject was last fall. It was published in Nature Materials because that work was capitalizing on a new material called ion-selective membrane, Ionophores. The test was done in frog sciatic nerve. So that's the first model used by Galvani in 1600s, I don't remember. So it's a very interesting and easy experimental model to test on anything neuroscience. And so it's well characterized, it's very easy to implement. Even we could do it. And it's an ex vivo experiment. So we took out the sciatic nerve from the frog leg and we artificially excite the nerve, and that has been done by Galvani 300 years ago. And then what we asked is the question whether if we can apply another coating to the electrode of these functional electrical stimulation electrode, can you actually decrease the threshold current to excite the nerve? That's the question we asked. So we compared the two situations where we would have a same nerve, but we try to excite the nerve with just a metal electrode. And then you need to achieve a certain pulse duration and pulse height in terms of current amount to be able to excite the nerve. Now, that threshold is well defined for each nerve. Now we switch the electrode and we actually have a new electrode. It's the same metal electrode, but now coated with the calcium ion selective membrane material. So this material transfers or conducts the calcium ion preferentially. And what it does is actually deplete the calcium ions around the nerve. So you actually make the calcium concentration locally go down and then try to excite the nerve that way. And then we were clearly seeing the decrease of the threshold current. These implants are implanted in vivo, perhaps with the batteries connected to that. So if you can lower the amount of current necessary to excite the nerve, then you can make the battery last longer, and then you may be able to avoid the unnecessary surgery. That's the one benefit. But I think the more important benefit is the fact that the electricity is actually conducts everywhere in the tissue in our body, because our bodies are conductors. And so the bigger challenges in prosthetics is that you try to excite a certain nerve in order to move the arm, but then because the electricity required to excite the nerve actually can conduct to every other place, it can excite the other nerve, maybe some sensory nerve passing by, and it may create some pain because of that. It may create some heat because you are japping too much current into the system. So those parasitic activation, parasitic current issues are actually bigger, and I think we can actually make a clear impact on that because we can potentially lower the amount of current significantly.